So this is our 14th week of multi-scale modeling. Today we're going to be continuing to talk about network model integration with cellular modeling. I'm going to review some of the things we did in a hurry last week. So we'll start out with the models that we were working on last week. And then I will ask you to work through a little bit of a different model. We'll see how that goes. And we'll stop on the network modeling early enough that I could do some introduction to compartmental cells. I don't know whether we'll actually get to do an exercise on compartmental cells. And I should say from the beginning that the slide deck on compartmental cells was developed by Professor Giulio Belmonte at North Carolina State University. Uh, he he's the expert on that, and he has a great slide deck. The slide decks I'm going to be showing is a shortened version of the one he uses. If you want to see his lecture version of it and the full slide deck, those are available online. As always, I need to remind you everything is live streamed and recorded. So we're going to start by rerunning the delta notch simulation. We didn't really get the steering panels implemented last time, so I'm going to ask you to do the steering panel implementation. You'll need that for your project, so it's important that you're able to do that on your own. You can't really do the projects without it. I'll talk briefly about the print window, which I think we may have covered briefly before, but I'll mention it again. We may skip exercise 14.4 in the interest of time, seeing how things go. And then I would like to walk through the Zartmann model of calcium dynamics, which is very similar to the delta notch model. Again, we may or may not cover all of that today. Normally, I would assign that as homework, but I didn't want to add homework assignments when people were working on their projects at this point. And then I'm going to start, at least on the introducing the concepts of compartmental cells to you. It's quite a powerful technique. It can be a little bit finicky. And I have to try to get to this exercise 14.9 of creating an epithelial layer. Although actually running that, we may have to wait till next time. Okay. So are there any comments or updates that people want to give me about their projects at this point?
I have one.
Sure.
So last week I was trying to develop something in this Joshua simulation with Alex and Emma, and we were annoyed about the time it takes to run the simulation, and I'm afraid we will not have enough time to replicate the results using his simulation. And we were discussing about switching to a simpler simulation to get the results done.
I would strongly discourage you from changing anything like that. Uh, when you say you won't have time to replicate, you mean you can't run enough replicates for different parameter values to be able to replicate everything that he did? Is that what you're saying?
Yes.
Then do what you can. Can you
Okay.
run one replicate simulation?
Yes.
Okay. How long does one simulation take to run?
About 1 to 2 hours.
Okay. So it's not going to be a lot of fun for somebody as a demo, but that's all right. Uh, you could report that, but as long as you could give people parameter values that they could use to explore it, that may be enough.
Okay.
I mean, uh, it's due in a week, so you can't start changing the thing you're doing at this point. Hayden, I don't know if you have any advice beyond that at this point. I would not change what I'm trying to do when it's due.
I would
You
agree with that.
you wanna stick with what you got at this point and do the best you can. If you can figure out a way to speed it up, great. If it runs slow, then we can't run 100 replicas. We could run 3 different things and show 3 different examples. I mean, the simulations that are in that paper were thousands of replicates, so it's not necessarily reasonable to expect that you would run that many replicates in the context of this course. Okay. On the other hand, showing you understand how the simulation works, and having a user be able to control things like the rate of spread of interferon or the rate of viral replication and see what happens, those are reasonable things to do. Okay. So I think it could still be quite a useful educational exercise, even if you can't run multiple replicates for each parameter value. Okay, William and Santoshi, do you want to say anything about where you are?
So sir, we have replicated a first model from the paper, and I think it's running well, but there are still a few models that we have to replicate, and it's kind of taking time.
Ideally, you want to make sure that you have something that you're solid on that you can hand in. So I think focus on getting one thing done and written up correctly, and then add additional material as possible rather than trying to get everything done before you start writing it up. Okay, good. Well, I'm glad to know that that you have working simulations. That's the key to going forward here. I want to come back to antibody SPML integration. Again, we've been through this now 2 weeks. We're going to continue to do it. The first thing you have to do to run some cellular models in the context of CompuCell is specify the dynamic network model. That model can be specified in a file or in, I guess I'm going to take this off. Look at it. That model can be specified in SPML or CellML, which are markup languages, or in antibody. It can live in a string inside the simulation or be read from an external file. So you have a lot of flexibility. We've only looked at specifying it in a string and then with antimony. Then you have to load that network model into the individual cells or into the simulation as a whole. And we talked about different ways of doing that. Again, I strongly remind you that there are manuals online for all of this and examples. There also are demos with this done in the CompuCell demos folder, not the demos for the course, but packaged with CompuCell as well. And then you have to be able to read and write antimony variables. Again, that's pretty easy because all of those variables show up as dictionary entries, but not the dictionary for the cell. It's cell.sbml.modeldict. And then we talked last week about the delta notch to couple variables between the cell, which we have to do essentially by hand in Python. Just reminding you of the syntax, add antimony model to cell is our basic function. That model string is reading it from a text string in the simulation itself, reading from a file. You'd say model file and then the path name. You have to give the model a name, so it's going to have a handle inside of Python. And then you can add it to a cell ID, a cell referenced by the cell handle, to all cells of a given type, more than one type, or a free-floating model as well. The only thing we haven't really played with much is the step size, which converts 1 unit of antimony time to 1 Monte Carlo step. And then typically, if we're going to say assign, read from the values. Again, the reference is cell.sbml. The model name you assigned when you loaded the model, and then quote, and then the name of the variable in antimony, or the parameter, or if you've named a rate law, the rate constant, the rate values as well. And so the one thing that you have to do is if you want to use a tracking field, you'll have to copy that value from the cell.sbml.model name dictionary into cell.dict value. And again, you can read out all of the values at once using the function .values if you want. Something that we did last time and that you'll need again today is iterating over neighbors. We have the iterator function for cell in self.cellList that iterates over all the neighbors of a cell. In this case, I very naively, I remember Emma had a more elegant way of doing this, a more Pythonic way of doing this by stacking a list and then using functions to average it. Here I'm doing the averaging by hand. For neighbor comma surface area in self.get cell neighbor data list, that returns pairs where the pair consists of the handle of the cell next to the cell's current cell and the contact area between the two cells. And that allows you to add up the common surface area, for example, and the number of neighbors. And then you can do averaging based on that. So in our delta notch model, we used the Collier model in which delta on a neighbor turns on notch. If you're my cell and neighboring cell, delta in my neighbor turns on notch in me. Notch in me turns off delta in me. That's the function. So it's a cross inhibition. And so we have the production of delta depends is inhibited by notch. That's the first term here on the right. We have a decay of whatever delta we have. Notch is only produced if there's delta in my neighbors, and it decays. Again, this is a very highly simplified version of this model. If you want to see a more elaborate version, there are many, many out there. Uri Alon, in particular, for example, has published some beautiful delta notch models. However, the basic functionality of the cross inhibition works even in the more detailed biochemical cases. And here we have the typical parameter values. These scalings, A equals 0.01, B equals 100. You could rescale this so everything is between 0 and 1, but that's the values in the source paper, so that's what we're using. And again, you can see that here, this is working in fruit fly. You see delta notch patterning. This is working in zebrafish. You can see delta notch patterning. In your stomach and gut, this is happening every day as the cells are replaced in the lining of your intestine. If you've ever eaten tripe, you've seen it in action. In the US, we don't eat a lot of tripe, and in a lot of places they eat tripe. And so this kind of patterning, it happens in human eyes as well, the rod and cone cell patterns. It happens in our blood vessels as they pattern. So this is one of the most important contact-mediated signaling mechanisms in biology, development and homeostasis. So the exercises that we did, what I'd ask everybody to get CompuCell going, and I'd like you to launch, if you can, launch the delta notch simulation from last time, and I'll give people a minute. Please give me a thumbs up when you've got it running, or type ready in the chat. To get this running, I'm just going to review this, what we did with people, but I'm going to assume that you've done it, but I want to walk you through it because we're going to do this again in a slightly more elaborate context. So again, what we're doing is we're creating a simple 4x42 by 42 simulation. In this case, periodic boundary conditions. I think, Emma, maybe you didn't put periodic boundary conditions in your simulation originally. That may have been, last week we were looking at why it looked a little bit different. That may have been one of the things. I wasn't sure. We're using volume local flex, contact energy, and neighbor tracker. You need neighbor tracker. Emma, I think you used surface area constraint as one way of forcing neighbor tracking, but there's a plugin called neighbor tracker that gives you that information without turning the surface area constraint on. And then we created 7 by 7 cells, set them to a target volume of something like 50 to the lambda of 2. Okay, so that's what that looks like. Here we've got dimension 42 by 42 by 1. run it for a long time. Temperature fluctuation amplitude of 10. Neighbor order of 1 in this case, which means somebody else wrote it because I usually use a slightly longer neighbor order when I do those. Boundary periodic in X and Y. And then we defined a single cell type, which we called cell, of a bare volume plugin. Center of mass tracking, we added that neighbor tracker plugin, which will allow us to return the identity of the neighbors in the area of contact. If you loaded the surface constraint, that would also do the same thing. And then we had the contact plugin, and we left that with the defaults. Then we used a uniform initializer to fill the entire area with cells of 7 by 7. Okay, and the next thing we're going to do is we're going to implement our simulation. In this case, I'm writing it essentially as ODEs. I could write it in a more networky way by separating the creation and destruction of the chemical into 2 separate lines. That would be more antimonian, just the way you have sort of Pythonic and procedural approaches to coding. There's an antimonian approach to coding and an ODE approach to coding. I don't know whether people care about style in mathematics, but you do notice it when you do it a lot. So d by dt, we write nothing goes to d. Nu times 1 over 1 plus b to the n times, to b times n to the h minus d. Nothing goes to n at a rate d average to the k divided by a plus d average to the k. Minus n. As always, we have to remember that we use caret inside of antimony rather than star star. We set those additional parameters, a to 0.01, b to 100, nu to 1, k to 2, h to 2, d to 1, n to 1, d average to 1. And notice the d average is actually something coming from the outside, and that's going to get updated every time we run, every time step. Then we're going to create tracking fields to keep track of the levels of D and N. In this case, I created one extra tracking field for D average, which is sort of interesting in its own right. And as always, that goes in the init function. So if you're looking through your twit, you'd be seeing these things. I'm just reminding ourselves how to do tracking. And then we're going to add, in the start function, we're going to add that antimony model. In this case, it's most convenient to add it by cell type because we only have one cell type. I called my model string delta notch model. You might type something shorter. I named the model DLN for delta notch. The cell types I'm going to do are just cell because I only had one cell type here, which I called cell. I defined the step size to be 0.2. And there's this. And then for cell and self.cell list, setting cell.lambda volume to 2, target volume to 49 or 50. And then I wanted to, if I start out with all the delta values or all the notch values exactly equal to each other, nothing happens. So I have to break the symmetry of the problem a little bit. So I make the initial values of D and N the random values between 0.9 and 1. The final configuration you get doesn't depend on that, as long as they're not all exactly the same to begin with. And then I copy the value from the SBML dictionary into the cell dictionary so that it gets displayed. Here, if I wanted to, I'd also copy the D average into the cell dictionary. Okay. Now in my step function, I first time step my SBML model. So that advances everything forward in time by 1. You could say, well, if things are very sensitive, maybe I need a smaller time step. Well, if that's the case, you effectively have to change by hand the value of time in the simulation. CompuSol doesn't do that for you for free. So in that case, that time step function, the step size function in the low antimony model would be made smaller. And then what do? I copy the current value of D from the SBML dictionary to the cell dictionary. I set celldict average to 0, the number of neighbors to 0. I iterate over the neighbors. I check if the neighbor is actually a cell which has something inside of it, because the medium doesn't. If neighbor, then celldict dav plus equals neighbor.sbml.dln of D, and add the count. I could easily also add the contact area if I wanted, the common surface area, if I wanted to normalize by the contact area. All right. And then I'd ask you to run it, make sure that it's running. Give me a thumbs up. And I know Emma already last week kept track of the contact area. And then when you're doing your average, weighting by the contact area, why doesn't everybody do that little extension? And I actually already showed you at the very beginning, I showed you the code for that. For that, I'm going to step back. If you look at the code that you have here, what do you have to do? You have to, in the middle line for neighbor, comma, common surface area, that's returning the common surface area. So you'd have to have a common surface area average equals 0 after nn equals 0. And then where you do nn plus equals 1, you'd have to add to that common surface area, to the total surface area, that common surface area. And then when you update the values of D average, you'd weight by the contact times the area. Okay. In other words, when you do celldict dot of D average, instead of plus equaling neighbor.sbml.dln D by itself, you'd multiply by common surface area. And then instead of dividing by nn, you'd divide by the total contact area of the cell. Okay. Why don't people try that? If there are any questions, let me know, and I'll give people 2 or 3 minutes to do that. You have to add 2 lines of code and change 1 line. Give me a thumbs up when you got it. I know Emma did it last week. If you've got it running already, then you could ask the question, what does it look, how is it different between the two? If you switch back and forth, does it look different or not? If the cells are all roughly hexagonal, so that they all touch each other with roughly equal area, it doesn't do much. In reality, sometimes the cells have rather complex shapes, so they touch some of their neighbors a lot and some only a little bit. In those situations, this can make a big difference to the pattern. Especially if you have parts of the cells that are not in contact with something with delta notch. So for example, with vasculature, human umbilical vein endothelial cells, they touch end to end, but they think of the segments of a water hose that touch at the ends. So the amount of the surface area over which they touch endothelial cell, the endothelial cell is pretty small compared to their total surface area. The delta notch signal is only happening on the ends, not on the sides. Okay, Alex, did that work for you?
Not sure. I got part of it, but I'm not sure.
You want to screen share it and we can go through it?
Sure.
Okay, so what you have there is the one we started with. Okay, you've got divide by common surface area.
Yeah, I missed what the other part
Right.
was.
Okay, so what you'll need to do there is actually, so that's not quite what you want to do. You don't want to divide by the common surface area, because then if two cells are in contact a lot, they have less signal, not more. So what you want to do is create something like n common total surface area or something like that, and make it 0 to begin with. Okay, good. Maybe 0.0 because you want to make it float. Okay, and now you want to multiply by common surface area instead of divide in line 104, because you're weighting it by that. Now you want to add common surface area to total surface area. And then in line 110, instead of saying if number, you'd say if total surface, because you don't want to divide by 0. And then you divide by average would be divided by total surface. Okay, try that. Okay, so it doesn't look terrible. It's not terribly exciting, but it's not, didn't crash, it didn't break your program. And again, this is a little bit more realistic in the sense that you really do have a signal that depends on the contact between the two cells. If the cells are touching a lot, the signaling is more effective than if they're only touching a little bit. Great, thank you very much. Okay. So we ran that simulation, and as Alex showed us, it doesn't look that different from the old simulation. There are a couple of things that I want to remind people of. One of them only works on PCs, or if you're running on a AWS, for example, it doesn't work on Macs, is one of the other nice features, at least on Windows of CompuCell, is the function called tile. If you pull down under the Windows menu in the player, there's the new graphics window that can also be gotten by hitting the little window stack icon under the tool menu pull down. But if you're not on a Mac, and this is not a CompuCell issue, this is a feature that Macs do not allow, unfortunately. There is a function called tile that will automatically resize and lay out the sub-windows that you've defined. And so that could be pretty convenient if you create a couple of windows and they're overlapping each other, you could hit tile and relay things out, and it makes things easier. But that, again, for some reason, is a feature that's not implemented. The core feature isn't available in Macs. It's only available in Windows. So on Macs, you have to lay out your windows by hand. Okay, I want to remind you about the steering panels. We went over those pretty quickly. Again, the point of steering panels, and in the example that Alex showed, there was a steering panel implemented already, but I want to go over them again because, again, it's important for projects. So steering panels let you change, especially Python variables, although you can also change XML variables if you want. Updating XML variables is a little fussier than Python variables. There are only two widgets implemented at the moment in CompuCell. It would be nice to have more. There's a combo box, which is a pull-down menu, select menu, and there's sliders. And so combo box lets you, for example, here I have a function called a killing type, and I can pull down select type of treatment, non-constant killing, So this is for selecting options, discrete options. And the slider allows you to select roughly contiguous values. You actually have to set the the resolution of the slider in advance so that it's not a perfectly continuous set of numbers. Again, if you haven't done it before, the steering panel uses event-driven syntax. Once you've done it once, it's not so bad, and it's all in the Twidit menus. But again, you're going to do 3 steps. You're going to create a steering panel window, which defines which parameters or variables you're going to modify and whether they're modified by a discrete pull-down combo box or by a slider. If somebody felt like extending CompuCell to have more widgets, that would be nice. But those are the 2 we've got at the moment. You're going to define a function that watches for panel updates and reports them. And then you're going to have to change the value of the updated parameters. And in general, all of these steering panel definitions go near the beginning of simulations before all the steppables. They go up at the top in the preamble before you start the class definitions for the steppables. So between the init and the start function, you add the def add steering panel. That'll pop up a steering panel. And that takes self as an argument. And steering panel doesn't have a handle associated with it, so you can only create 1. I suppose in principle you could create 2, but we don't have that option in this. And we don't really have much in the way of formatting the steering panels. If you were using full IPy widgets, you could do fancy layouts and things, but we don't support all the fancy stuff. So you have def add steering panel on self, and then you give that steering panel, inside of that steering panel, you're going to say how many lines or how many variables you're going to modify. So here self.add steering parameter, each line gets a name. Here's my parameter for a slider. Val is the initial value, the default value. Min val gives you the left-hand rate limit. Max val gives you the right-hand limit. Decimal precision gives you the resolution as you drag it. If you make that too coarse, then you can't maybe not able to adjust it enough. If you make it too fine, the system could get stuck trying to update too frequently. And then you have to name your slider. Each slider gets its own name. And it's a type. So a widget name is the type. In this case, it's a slider. So the first one adds a continuous slider. The second one, combo, adds a combo box, a pull-down menu. And in this case, we've listed all of the labels that are going to go on it: 10, 20, 30, 40, 50, 60, 70, 80, 90, 100. Those could be string values as well. They don't have to be numbers. So we first add that function def add steering panel, and then inside of that we add as many lines as we need. The default value, that first value, is the initial value before the user does anything. Min val and max value restrict the user's range of options. So you want to think about those pretty carefully. Sometimes people will have a slider that controls the slider limits for the other slider. You can do things like that. So the first thing we have to do when we're going to do this is we have to give that variable name by parameter slider a name, a unique name. Then we have to select whether we're going to use a slider or a combo box. We have to set the default value. If it's a slider, we have to set the minimum and maximum value and the decimal precision of the thing that was returned, how many digits are returned. And if it's a list, a combo, we have to make a list of the values that are going to be returned. So def add steering panel, self add steering parameter, my parameter slider, et cetera, et cetera, et cetera. When you're going to call it, and boy, that the font is a little bit hard, there's going to be a function called process steering panel data, which is called every time every time somebody touches one of those sliders or pull down menus. And you have to match, if you say called the variable my parameter slider in the definition, then when you're doing the update, you have to reference it exactly whatever you use there. So those two things have to match, just the way they do for dictionary key, or just the way they do when you're creating a plot window. When I do the update and get the value parameter combo in process steering panel, and I'm sorry, I'm not sure why the font color is hard to read here. I don't know what happened to that. It's there, but it's pretty gray. So you're doing so. Let me fix that. Give me a second. So no, it's too hard to read that. Sorry about that. I don't know what happened to that. Not sure why they're grayed out. Sorry. I noticed there are a couple of funny font things going on in here that I'm not quite sure where those came from. Occasionally PowerPoint will decide that it gets unhappy about fonts. I notice in the title slides, in the title section of some of these slides, the font spaces. Okay, so this should be easy. So in the process steering panel data, this is called every time anybody touches the steering panel. The first thing you do is you pull the value from my parameter slider, and that returns the number on the slider. That number is local to the function here, and so you have to move it. In this case, for example, if I want to update the target volume of the cell, I'll loop through all the cells in self.cell list and update with them with a new value from that slider, or update lambda volume from the pull down menu. So in this case, every time somebody uses my parameter slider, it's going to change the target volume of the cells. Every time somebody uses the pull down combo, it'll change the lambda volume. And so that's how these things work. What's a little counterintuitive is all this definition of both the slider panel and its processing happens outside of all of the definitions that you're used to. You're used to working mostly inside of the class. Here you're outside of that. Okay. And I mentioned before that you could use this, for example, to have the simulation start and stop. If you use a pull down saying select simulation to run, and another pull down that says pause simulation, and then within the step function, you say while self.get steering parameter simulation type is select simulation to run, pass while self.get steering panel type pause simulation pass, then the user can actually start and stop the simulation by selecting those buttons. Again, that's something that you're not going to use if you're doing production code for a study, but if you're doing something that you want a user to run and say, see what happens when you do this, that's a very convenient feature. So I think a lot of you have already done this, but I would like people now to take the simulation code that we were just working with, and the example we went through, and add the steering panel code to change the values of A and B in the antimony parameters.
think you might have lost the screen share at some point, James.
That's bad. How far along back was it?
Just a couple of minutes.
Okay. Well, where was it?
I think it was just the last two slides.
So this from here?
Probably somebody else maybe could help me out.
Alex, if you were watching this, when did the screen share die?
I think we saw that one. I think it was whichever one you started editing, it might have gotten lost.
Ah, okay. So after I fixed the form, the font, it here probably.
Mhm.
Okay, sorry about that. So apologize. I had stopped the screen share because I wanted to. These were the def process steering panel data function was grayed out, and the self.get steering panel parameter was grayed out, and I wanted you to be able to see that. So, okay. So again, let me come back to this. You create a function that creates the steering panel and defines what the widgets are in the steering panel. You create a second function called def process steering panel data that is called every time the user touches the steering panel. And within that, you read the updated values of each slider or each pull-down menu in the steering panel. So if the user has touched one of those widgets, it will return the new value. If the user has not touched one of those widgets, it will return the old value, the previous value. Once you've read those new values, you have to use them because they're not persistent coming out of the function. And so in this case, we're reading the value of the new target volume for the cells, the new lambda volume for the cells. We're iterating over all the cells in the cell list, and we're updating cell.target volume based on the new value of the myParameter slider widget. And we're updating the cell.lambda volume based on the new value of the pull-down myParameter combo widget. And then I was mentioning that you can use this with two feudal loops to allow a user to start and stop or start and pause your simulation as it's running. Of course, you can always hit stop and you can always hit step or pause. But if you have an instructional video where you want the user to stop the simulation at particular times, or you want the simulation to stop automatically and wait until the user does something, you can use this kind of functionality with pull-down menus to allow the user to see what you want them to see, and then make the user hit a button to restart the simulation and continue. There isn't, I believe, an obvious way to do a pause from inside of the simulation. I may be wrong on that one. There may be actually a pause simulation function inside of CompuCell. But in general, well, it's not something we do. In this case, for example, if you want to force the user to select which of multiple options in the simulation they're going to use before the simulation starts, you can do it this way. And there's a NanoHub cancer evolution demo that uses that functionality that you can try out. Okay, so the next thing that I'd like you to do is to, and I know, again, some of you have the code that's already doing it, in which case that's fine, is to change the values of A and B in your delta notch simulation. And so to do that, you're going to create a steering panel. That steering panel will have two widgets, one of which controls, they'll both be sliders. One of them controls A, one controls B. You know the initial values of A and B because you've seen them from the old from the antimony that you have now. So the default values should be the ones that you used already. And then you're going to have to set the range of allowed values of A and B. They can't be less than 0. You'll have to decide how big you want them to be. Probably 10 times the initial value is reasonable. And then you'll have to set the resolution of it. You'll have to create that process steering panel. And when you do that, you'll have to loop through the cells, and you'll have to update the values of A and B for each cell. Okay? And I'll give people a few minutes to walk through that. The completed code is available in the demos. If you've already done it, I'd suggest trying different values of A and B and seeing how the simulation differs, because you'll find that the delta notch patterning changes a lot when you change the values of A and B. For some values of A and B, you get no patterning. For some values, it's quite strong. Sometimes we get oscillatory patterning. Sometimes you can get delta and notch to vary together, sometimes in opposition. And if you've done that and you're done with that, you could try some games with the coupling strength between neighboring cells, for example. Or you could use a combo box to select whether you normalize the simulation by the number of neighbors or by the contact area. Those are all things you could do inside of this.
Is there a vending machine in the building?
Is there a vending machine in the building? Don't know.
I think there's one on the ground floor or the basement, but I remember seeing one somewhere.
I hope she's not locked out coming back in. I think the door to this aisle may lock at 5 o'clock. 5:30. And again, give me a thumbs up when you've got it working. If you're using the one where you've already got it working, that's fine. Just tell me. And as I say, so Alex says it's working. There's quite a lot to explore. There's quite a lot to explore, and so you can try different values of A and B and see what happens. See if you can find a value of A and B where you get no patterning at all, where the values become essentially constant, uniform values of delta and notch. See if you get one where delta and notch both go high together or both go low together rather than high delta, low notch, low delta, high notch pairing. And see if you can find one where they oscillate, where you get bursting patterns. If there's only one cell type, it's somewhat limited in terms of what you can do. If you have two cell types, then you can play more interesting games, because you could have one cell type be larger and one smaller. And if you have larger and smaller cells, then you get very different delta notch patterning. Or you could have the parameter values be different in the two cell types. It's also possible. Santoshi? You want to show it?
Apparently there is a error in this line. The cell one is not being called.
So, sorry, what did you start with? What was the code you started with? Where did this code come from?
I've tried adding from the old CC3D.
So you said you had the simulation running before,
Yes, sir. But once I've started with this one, I
but
didn't.
you wouldn't be changing anything there at all.
Yeah.
So what did you change?
The cell one.
No, you wouldn't change, but that's in the old. So what was the simulation that worked? Why don't you show me that?
That's the okay. That is before like adding this one. I've just started with the like with the starting simulation with 42 into 42 ladders. That's what
Yes.
it has worked. But once I've added this SBML, that has been the error.
Oh, so you didn't have the simulation working?
Yeah.
No. Okay. So could you please load the demo code, exercise 842 delta notch steering?
Yes.
Okay, so it is important. It is important that if I ask you if it works and it doesn't work, you need to tell me. Because I can't help you if you tell me that it works and it doesn't. And since these are progressive exercises, if you get stuck and it doesn't work, then you're not going to be able to do the additional exercises. Okay, thank you. Okay, so we're going to keep going. So again, to implement this, to create the steering panel, after the init, you say def add steering panel, and we're going to add two widgets. We're going to add one called A, which has an initial value of 0.01, minimum value of 0, maximum value of 0.02. Could maybe be a little bit bigger. Decimal precision of 3 digits, and it's going to be a slider. We're going to add another one. We're going to call B. Initial value is 100. It goes between 0 and 200. Decimal precision of 0. And the widget name is slider again. So we have to make one change basically to the boilerplate code that Twidit gives us there. And then we have to process that. And so we have in the def process steering panel data, we have to read the value of A and B from the steering panel. So we'll get say self.get steering panel parameter. The screen share is working, right, Hayden, at this point? Yes, I hope.
Yeah, looks good.
Good, okay. We have to read the values of A and B, and those are local now to the function. So then I have to add a loop over my cells, for cell and self.cell list by type of self.cell, and then I update the value of A and B from the values pulled from the steering panel. And that's all I have to do. So I don't change anything inside the start function to the step function. Everything that I'm changing is now in the init of the simulation. And did you find, Alex, when you ran that, did it actually change the pattern at all?
When I made the B really low, yes, but otherwise not really.
Okay, when you make B really low, what happens?
Everything just becomes red.
Okay, so it blows, right? Either one of the inhibition doesn't work or the activation doesn't work out. There are games you can play to make it a little more interesting. It's a little bit finicky to do it, but that's the kind of thing where For example, if you're going to do that as an exercise, homework exercise or an exercise for students as a demo, you'd want to give them some guidance about appropriate values of A and B. And then you'd ask them why. What is it about those values of A and B that give different patterns? I wanted to talk about something else that we use primarily in the context of educational or user-facing software. It's not very sophisticated. By default, CompuCell will display either to the Python console, which is not something you see. There's an option in Player that says to redirect the console output to that window at the bottom of Player that displays the error messages, but it doesn't work. And so you can pop up a message window, self.message window equals self.add new message window, and then the title of the message window. You can have more than one of these, so you can define the handle for them. And then using self.message window .print, you can print to that message window. And so now, if you're thinking about writing a user-facing application, you can display instructions. Do this now, or this happened, or be aware of this, or change this value in a window that the user will see when they run the simulation. It's pretty limited in terms of what you can do in terms of formatting. You can change the color of your text. You can make it bold, italic, or underlined. And you can clear the window if you want. So there's a clear function. Occasionally, when you try to print a lot to it, the buffer will overflow and it'll crash. And I'm not exactly sure what causes that. I think that's pretty well fixed in the current version of CompuCell. But you wouldn't want to display, you wouldn't want to put an enormous amount of text into that window. But if you want to communicate with the user, the simulation runs for some amount of time, then it pauses, and the user has to hit a button to resume it, and they're supposed to notice something happened. This is a good way of communicating. So it's not a very highly developed feature, but it does allow packaged applications to be built a little bit more conveniently. Why don't people try in the code that they have now, just adding in the start function, add self.message window, self.add new message window, title messages, and in the step function, print the time. And then here I've showed style equals bold, italic, and da-da-da. You're only allowed to pick one of those. You can't have all, there's options. Just try that and see if it works. Emma.
What, if any, are the advantages of these text windows in opposition to just having things printed in the terminal? Is it mainly for usability by people who are programmers, or?
Well, so the terminal window is not always visible when you're running Player, right? The terminal window is the Python control window.
But I see that I can put print in my Python code and it will, but I just have to.
Right, but it doesn't display within the Player, within the frame of Player. It displays into the console. Now there is in Player a setting that says direct console output to Player window, but it doesn't work. And I don't know why it's hard to fix that, but apparently it's very hard to get that to work. This is a little bit of a hangover from the NanoHub era. In NanoHub, the console is not visible. NanoHub does not, when you run Player and Twit in NanoHub, the Python console window is not visible. They do not allow you to type general Python and execute it in the Python control window. And that's not something that we had any control over. In other words, that was a restriction of the public server. They didn't want you executing free code Ironically, I mean, you could type free code into a steppable and it was okay, but they wouldn't let you run free code in the ID directly, in the Python shell directly. But again, this is more if the person is supposed to launch it and run it, and it's supposed to be a freestanding application. The idea is everything that the user has to interact with is within the box of the player window. But it's not, again, for people who are more sophisticated in terms of user, I don't think you gain a lot from it. But the only other thing that you gain perhaps is that within that normal Python console, there's a certain amount of junk that gets printed by default, right? The time steps, how many cells there are, how many updates, and so on. And so it could be a little bit difficult to find things. And so this is a little bit, again, it's really there for hand holding for a user-facing application. All right. Did it work when you tried it?
I'm still.
Okay. This one is not, as of the previous, I don't know if it's intuited yet or not. This was not this was one of the few things that was not implemented. It was not a twitted code snippet, mainly because Maciek didn't like it. He didn't want to do this. I had asked him to do it for years. He didn't really want to implement it, and so he was a little bit grudging. I'll edit this out of the video, but he was a little bit grudging when he put it in. So he didn't really want to encourage people to use it. But for example, if you're going to run CompuCell in its Jupyter Notebook incarnation, you might want to use this as well, because within the given cell of the notebook, it'll pop up the player windows, and you may not be able to see the console output, depending on how you configure it. Did it work? Yeah. Did it crash on you when you ran it for a long time?
How long is long?
If it hasn't crashed now, it'll be okay.
Well, I'm trying to do funny stuff with it, so.
Okay. You're poking at it to see. Okay. I mean, it's very basic, but you could actually do, again, if you want to tell the user do this now, it's helpful. Or you want to print the value of a variable, for example. It's helpful. Anybody have problems with that, or did that work? So here was an example of that. In the start function, I added self.message window equals self.add new message window, called it messages. Then in the step function, every 10 Monte Carlo steps, I go through all my cells and I print the cell ID, the value of D average, and I put it in italics in blue. So every 10 steps, it gives me the value of the neighboring average cell signal, delta signal, and prints it out. Again, you could say, well, I can read that from the tracking field, but the tracking field will give you an idea of roughly how big or small something is, but it won't give you the actual numerical value very accurately. And so if you want to know what the value really is numerically, you might want to print it out. So that's an example of that. Again, there is this annoyance that sometimes if you write too much data to the text window without erasing it, it could crash player. That seems to depend on the computer you're running on, so I'm not sure what causes that. Okay. And I think, let's see how we're doing on time. I was going to ask you to do an exercise where you use delta and notch to control the cell growth. I think we're going to, in the interest of time, we're going to skip that today. I'll enunciate the exercise with you, but I'm not going to have you do it here. I was going to make it a homework exercise, but again, I think it's more important that people work on their projects. But let me just walk you through it for familiarity. I'll remind you that in the simulation that we have running now, we do not have cell growth and division. We don't have the mitosis steppable loaded. If we're going to add a steppable, we could do that in a couple of ways. The easiest one is to select SBML delta dot steppables, and then the add steppable function here. And then when we do generate steppable, we have choices of generic mitosis, cluster mitosis, run before MCS. The one that we need to pick is going to be mitosis. And if we wanted tracking fields, we could actually tell it to paste the tracking field in when we create it, if we don't remember how to do that ourselves. And the exercise that I was going to suggest is have, why don't we just do one? Well, no, I don't think I want to do this exercise. I was going to say we should do it in a simpler way, but I don't think maybe we can have a vote. Emma, have you already done this exercise? Do you want to do it? So if you start out with a single cell or two cells, and then you have the growth rate of the cell be determined by notch. So if notch level is low, the cell grows very slowly. If notch is higher, the cell grows faster. When the cell gets to its doubling volume, it divides. That gives rise to a rather interesting patterning, because of course, when you're a single cell, if you only had one cell, there's no signal, and so nothing happens. That's why you have to start with two. But if you start with two, you'll get one high cell, delta cell, one low delta cell, one high notch cell, one low notch cell. The high notch cell will grow. When it divides, one of its neighbors will be suppressed, and then you'll get a layer of low notch cells around the high notch one, and so you'll get sort of a checkerboard that's having to continually update itself, because you'll get black cells next to black or red next to red. They don't like each other. One will win and force the other one, and so you'll see that the pattern will spread, and the initially high notch cells will become low notch and vice versa. But I think that may be a little bit too much to do in class now. But I'd encourage people to try that out. It's a fun exercise. And so to do that, and if people want to try doing it, it really takes about 5 minutes to do. You change the CC3DML uniform initializer to only draw two cells. That only takes a second. Just change the size of the box to draw. So why don't we do that? So first change the CC3DML to just draw two cells. Run the simulation. Make sure that works. Then use Twitter to add a mitosis steppable. You can call it growth and mitosis. When you do that, you'll have to edit the steppable, so the cells grow at a constant rate of 0.1 voxels per MCS and divide when they reach a volume of 50. To begin with, don't have the growth rate depend on the notch level. Just have them grow at a constant rate. That already gives you interesting patterning. And the templated code that comes when you add the mitosis steppable will try to switch the type of the cell from type 1 to type 2, so you have to remember to delete that or it'll crash. So you really only have to do two things. You have to change uniform initializer to draw a box with only two cells to begin with. Then you have to add mitosis steppable and you have to edit mitosusceptible to change the growth rate of the cell to make it slow enough, and then change the doubling volume, and then get rid of that offending 3 lines of code which change the cell type. And the other thing you probably want to do if you're going to run this simulation is I would make the simulation bigger. Instead of making it a 42 by 42 lattice, I'd make the lattice as a whole 100 by 100 so that there's a little bit more room for the cells to grow. Otherwise, you run out of space pretty quickly and the pattern isn't as interesting. One thing that I think is important, but at this stage of the class, is that it's important that people be at a stage where you could actually explore on your own a little bit. These exercises in class are really to show you sort of principles of operation of some of the functionality in CompuCell. And the hope is that you're able to master those enough that you can play and explore the capabilities of the package. And this is a place where that this would be definitely of interest. For example, if you're studying the growth of the eye, in certain species, the eye grows continually repatterns. For example, in frogs. As the animal gets bigger, it generates new retinal cells, and those retinal cells will continue to pattern with delta notch as the animal grows throughout its life. And so this is not just this is a little bit of an abstract exercise, but actually the process we're modeling here is one that actually happens in the developing embryo. Hey, Alex, how's it going?
Good.
Do you want to show it to us?
Sure.
Okay. Do you want to start it over so we could see what happens with the initial pattern? So, right. So initially, let's check the colors and see the delta and the notch. And you'll have to hit play again okay. So initially they're all high delta. And now as it grows, so why are we not getting any delta notch patterning? Huh. So the one you showed before had pattern.
Yeah, it just takes it a little bit. I'm also not sure why it's not updating, and I'm going to do it manually. It was working before. There it is.
Okay. So when you started it, did you randomize the values of delta and notch in the cells? In the start function? Yeah, you did. Why don't we make that there? I don't understand why it doesn't pattern earlier. That's a little bit surprising to me. So in this case, what you would have to do really is slow down the growth. Basically, the cells are growing so fast compared to the patterning time that you're not really seeing things. So maybe slow it down by a factor of 10. Yeah. And let's try that. Okay. Huh. Are you waiting the signal by the contact area, or is it by just by the number of neighbors? It is by common surface area. Okay. All right. So we may have to wait until there are more cells for it to start patterning. Huh. I'm a little puzzled why that center cell hasn't switched. That's interesting to me. Did anybody else get it to work, or Emma? How about you? Is it also doing nothing interesting, or is it more interesting for you?
Well, I might not be the most astute observer of these sorts of things.
Let's take a look. Let's see.
Pattern is much more distinctive than it was before I put this.
Okay, Emma, do you want to give us take us? Okay. Anybody else? I mean, this kind of exercise, it tends to be, you have to play with them a little bit to get the parameter values to work. This is what I did in terms of just having the target volume increment, having the cells divide randomly. And I got something that looked like that. So it patterned pretty quickly. So, but I think I wasn't using the weighting by contact area. So it's possible that the issue is that if you go back to by the number of neighbors, it may work better.
Are you meaning the screen share right now?
You want the screen share back?
I was saying, no, I know you were meaning to. I don't know if there's something I was supposed to be seeing.
Oh, sorry. Okay, so Emma. Okay, let's look at Emma. No, so I wasn't. Okay, so I lost the screen share.
Just that these panels don't close after the simulation ends. All right.
Yeah, I wouldn't recommend closing outright that steering panel. I don't know what that does to those functions.
The new ones are here. Those are old ones.
Oh, okay. All right. Well, that should be a ticket. Okay. So in your case, it definitely, as it's developing, you see patterning happen. So Emma's simulation is more or less what I would expect. In particular, since if you're a high notch cell, you don't want to be next to another high notch cell. If you're a high delta cell, you don't want to be next to another high delta cell. But a high notch or high delta cell divides, one of the daughter cells has to flip type. And so that's pretty nice. Okay, it's very interesting. The next thing that you might want to do if you want to play with it is have the growth depend on the notch concentration.
Oh, it does.
And so this one you have okay, so the high notch cells are growing faster than the low notch cells. Okay. Great. So yeah, that's a nice way to do that. And so then you're forcing on division that you're always going to have high notch next to high notch, and then the pattern has to update to accommodate that. Great. Okay. Thank you very much. And thank you, Alex, for saying that right away that the screen share wasn't taking. So that was helpful to that. It's embarrassing to talk for half an hour and then be told, oh, the screen share wasn't there. Or I was sharing the wrong screen. Okay. So this was code that I wrote. to do that. And this was the kind of output that I got. And again, that code is available to you if you weren't able to get that done in class. And then this was the second part of that, where we had the the growth rate depending on the notch. Emma made the growth rate linearly proportional to notch. It's sort of interesting if you make the growth rate proportional to square of the notch level. Instead of linear with the notch. Since notch basically goes between 0 and 1, squaring it slows things down a little bit. But that's actually rather nice. And so you could play with different functional forms of dependence on delta and notch and see how the pattern changes. And you'll see you get rather, it may not be so easy to quantify how they're different, but if you watch a number of simulations of this kind with very linearly proportional to delta and to notch or quadratic in notch, and then you can also try the other one, which is have the growth rate proportional to delta. Remember, the delta cells tend to be isolated. The high delta cells tend to be isolated. The pattern, maybe Emma, you could try switching, instead of having the growth rate proportional to notch, make it just change one line to make it proportional to delta and see whether the pattern changes. Again, you'll find that all of these things do occur in different biological settings. So I think what's interesting to me about delta notch is that I don't know of an alternative mechanism that behaves the same way. In other words, in a lot of situations, there'll be some molecular signaling pair that is used that you see occur in particular contexts, but there will be other signaling pairs that apparently are functionally equivalent in other contexts. The idea of a growth factor and a growth factor receptor, there are many growth factors, FGFs, VEGFs, and so on, all of which have sort of similar patterns of promoting growth or promoting differentiation or promoting motility. Even though they're different molecules, there are other molecules that have similar functions. In the case of delta notch, this contact inhibition mechanism, there must be some other molecular pairs that do that, but I don't know of any that are particularly commonly used. There's one that has a sort of a similar. It's pretty common to have, for example, for cell adhesion molecules, they're typically homophilic. They want to bind to a partner of the same kind. Delta notch is unusual in that delta on one cell binds to notch on the other cell. The other molecular pair that's common like that is F and ephrin. And in that case, F and F bind to each other and stick. F and ephrin bind to each other and stick. F and ephrin actually actively repel each other. And so F and ephrin, if you have a group of cells with F and a group of cells with ephrin, they'll physically pull away from each other to build gaps between tissue domains. And so, for example, in the formation of the somites during segmentation in early embryogenesis, F and ephrin separate the cell type, the cells. They actually, if a cell with F and a cell with ephrin come and touch each other, they actually don't just not attach, they actually actively pull back. Although interestingly, if you turn off F and ephrin, you still get the same pattern. So this is a case where the F-ephrin mechanism reinforces the patterning and it cleans it up, so the pattern is cleaner. But it's not necessary to have it. Semaphorins sometimes behave like that too. Those are all molecules associated with cell migration and cell. So semaphorins and epinephrine actually are important, for example, in neurogenesis, where when the motor neurons grow out between the somites to innervate the body from the spinal cord, they grow out, and then they spread between the somites. And the pathfinding there is dependent on those molecular species. I'm not a big expert on axonal targeting, but that's so that's not in the brain. That's on the other side, an innervation of myofibers. Okay, great. All right. So make sure my screen share is okay here. Now I would like to spend the next 45 minutes. I don't want to use up the whole class on this. This is a slightly more complicated model, and in this case, I'm going to ask you to download the model from the student repository. It'll be in the demos from last week rather than, I didn't create a new demos folder for the exercise, but the exercises should be here. This is a model that we developed in conjunction with a team at Purdue University. It's a model of calcium signaling in Drosophila and fruit flies during wing development. And if people have studied insects, they know that insects go through something called metamorphosis. You have the larval stages of the insect, and then there's a huge reorganization of the cells in the insect. Almost all, not almost all, but a substantial fraction, more than half of the cells in the larva will die, and their nutrients will essentially be consumed by other cells that will form a new embryonic structure in the inside the larva, which will then emerge as the later stages of the organism. Depending on the insect, there may be multiple metamorphoses during development. It's a little bit creepy in the sense that if you watch a movie like Aliens, where you have something that's growing inside of you and eating you, that's normal insect development. Insect development is sort of strange. So the reason I'm explaining this is that the term imaginal disc may be unfamiliar if you haven't taken a course in developmental biology. In insects, you have these clusters of cells within the larva that are called imaginal discs that are going to form the adult organs. And those consume nutrients from the surrounding tissue, they destroy the surrounding tissue, grow, and then replace the initial tissue. There are other insects aren't the only kind of organism that grow in this way, but they're the classic example of it. In the imaginal disc that's going to eventually form the wing of the animal, there are patterns, and these patterns are mediated through calcium signaling. And if you've ever done a study of, again, if you've read a biology textbook, one of the things that's a little bit less mysterious now than it was 30 years ago, but you'll see in the textbook, it'll say something happens and then there's an influx of calcium into the cell, and the cell does something very specific. And so one of the big mysteries has always been if you have calcium coming into the cell under all these different situations, how does the cell know what to do? How does it do one thing versus something else? A calcium influx in one cell does this, a calcium influx in a different cell does that. Why is the response to calcium important and different? And that's often known as the second messenger problem. And the answer is that calcium is basically a general signal saying do something. and do it in a pulsatile way. And exactly how it's excited and how it's responded depend on the other things that are going on in the cell. In the case of many cells, if you're used to neurons, then neurons fire things called action potentials, which involve ionic transport between the inside and the outside of the cell. Calcium is a little bit different in the sense that the cells have organelles inside of them called the endoplasmic reticulum. And during normal situations in the cell will actively pump calcium into the endoplasmic reticulum. And so it will reduce the amount of calcium inside of the cytoplasm and put it in storage, often related to calmodulin, inside the endoplasmic reticulum. And then when there is an influx of calcium into the cell for some reason, there are what are called calcium-gated calcium channels. And again, if you're used to neurons, this will be a familiar concept. You'll have voltage-gated or ion-gated ion channels. The calcium-gated calcium channels will open and then release the calcium that's stored in the endoplasmic reticulum into the cytoplasm. Cytosol would be correct, technically correct. And so the result is that an influx of calcium from outside then triggers a release of calcium inside the cell. And so an initial blip of calcium turns into a big spike of calcium. And then these pumps will then keep operating, and then we'll put the calcium back from the cytosol back into the endoplasmic reticulum. So you see small influx of calcium from outside, massive spike of calcium released by the endoplasmic reticulum, and then gradual decay of that spike due to pumping of the calcium back into the endoplasmic reticulum. This is very similar to what happens with an action potential with sodium or potassium currents, with the difference that instead of pumping between the outside of the cell and the inside of the cell, is in an action potential, you're pumping between two compartments of the same cell. And there are a number of molecular species involved here. IP3 and Circ-A are the ones that are most common. And with calcium, there are couplings between the cells through what are called gap junctions, which are basically holes between the two neighboring cells, which allow molecules to go across between two cells. They're not that big, so they don't allow large molecular species through, but calcium can get through. So there are gap junctions that allow leakage of calcium from one cell to another. So if you have a calcium signal into one cell, that signal by itself wouldn't be big enough to activate the neighboring cells. But the spike of calcium released by the endoplasmic reticulum is big enough that it's able then to trigger the neighboring cells. And so what you'll get is something happens and turns the calcium spike on in one cell, then that calcium will leak to the neighbors, they'll spike, they'll trigger the next cells and the next cells and the next cells. Again, if you're used to action potentials and neuronal activity, this will be pretty familiar, although the time scales over which this happens are slow. If you look at Zartmann's experiments here, it's probably pretty small, but the time scales here is 0 to 60 minutes. Whereas if you were talking about action potentials, that would probably be 0 to 60, not even 0 to 60 seconds. It would probably be 0 to 60 milliseconds that would be happening. So this looks a lot like action potentials, except they're playing out over a much longer time scale. Okay, and so Jeremiah Zartmann at Notre Dame is one of the world's expert on this particular kind of calcium signaling. And he's developed some beautiful computer simulations of it, as well as incredible experiments. And so he wrote a little model of the calcium-induced calcium release, which is based on this, where he effectively says that and I'm not going to go through the equations in great detail today. They're here and people can look at them. There is a pump that is continually moving calcium out of the cytosol into the endoplasmic reticulum. There are calcium-gated calcium channels that when the calcium level gets high enough in the cytosol, they dump all of the calcium out of the endoplasmic reticulum back into the cytosol. And there also are signals that control IP3, which modulates all of this in the cytosol, based on extracellular signals as well. And so that's the basic structure of the model, which was based on all sorts of experiments done by Jeremiah and others. And you have the functional forms here, which we could go into. One of the things that's sort of nice about this, and again, for today, we're going to have to take some of these things on faith. It's worth reading his papers, which are very elegant. But one of the things that's really impressive is he's actually been able to measure in these kinds of simulations that we're doing with molecular species. It's usually pretty hard to pin down all the rate constants. How fast are things reacting? Biochemically, it's pretty easy to measure equilibrium concentrations of things. But the equilibrium concentration gives you the ratio of the forward and backward rate constants. It doesn't give their actual values. And in this case, Jeremiah actually has been able, and some others, have actually been able to determine the actual rate constants for almost every chemical species in this rather complex reaction. So we're going to look at this model. And again, the basic idea is that we're going to have cells that have calcium-induced calcium release. If the cells are leaky enough, then they're actually going to keep spiking. The calcium-induced calcium release will be slow enough that it will induce another spike and then another spike and another spike. And there'll be other cells that are just below the threshold for oscillation. But if they get triggered by calcium from somewhere else, they'll spike. And again, if you're used to neurons, you'll have spontaneously active neurons or spontaneously bursting neurons. And those can then induce other neurons that are just below threshold to spike. So it's playing out. And the reason I wanted to do this was really for Emma, because I know you've got this strong interest in neural activity. And so I wanted to show you an example. It's not exactly neural activity, but this is the kind of model you could use if you wanted to model propagation of neural activity. I don't have a neural activity model worked out, but we could almost take each component of this and pull a Hodgkin-Huxley equation in and sort of map piece by piece. In that case, you'd have synapses, so you would, instead of the gap junctions, you'd have synaptic transmission, but the model would look very similar. All right. So there should be in the folder from last week an exercise 4_1_10_Zartmann_simple. Can people try to download that and run it? If you can't find it, let me know. If it doesn't run, let me know. Santoshi, do you have it?
I'm downloading it, sir.
William?
I always have issues getting into that Google Drive. Every single time it tells me there's a 404.
Huh.
I don't remember how I've done it in the past. I think certain things work, certain.
Hayden, do you have the link to that drive that you could share to the chat?
Yeah, let me go grab it.
The one on, I don't know. I've noticed that Canva sometimes appends things when I copy. It's not fun.
Oh yeah, you should only use the canvas links. Let me just get it fresh for you here. And then there's the usual issue that there's the usual issue that when you download it on a Mac, the Mac will unpack it, which is not what you want. So then you may have to put them back into a folder. Let me hmm. Oh, I see.
I just posted it in the chat.
Okay. Does everybody have it? Did it run? William, Santoshi? Okay. This is using that simple code we used originally. For the to run arbitrary antimony models. So that particular delta Dutch model, that's a delta Dutch, the particular calcium model that Zartmann wrote here is implemented in antimony. And if you run it here, you should see it plot a pulsatile time series where you'll see that P stands for the level of IP3 in the simulation. The spikes of calcium are a little bit less obvious, but the spikes of the IP3 are the P value you'll see spike up like that. So does that work for you? Okay. So now if you go into your Twinit, and this is the kind of place where it really would be nice to have a slider, so I don't have to have you edit the code. So if you go into line 24 of the code, and you'll see VPLC equals 0.5. That is effectively the positive feedback strength in the signal. So originally VPLC, let's see what was the original value. The original value is 1.5. And if you now make it 0.5, see what happens. If you make it 0.5, does it still oscillate? Spikes once and then stops, right? Depends a little bit on the initial condition. Okay, here's what I have. So, okay. So in my case, when I explored it, I actually did a binary search. If VPLC is less than 0.773, it doesn't oscillate. If it's bigger, it does. And actually, so you get some sort of interesting behaviors here as you change VPLC. You could check on, for example, what the frequency of oscillations is. And you'll find that if you reduce VPLC, as you make VPLC smaller, the frequency of oscillations goes down, or similarly, as you make VPLC bigger, the frequency of oscillations goes up. And the peak height changes as well. And one can actually develop what's called a bifurcation diagram, which we're not really going to go into here. where there are two different two different bifurcations. The one at 0.773 that we just looked at, and then at 3.75, there's another one. So for VPLC below 0.773, you reach a steady low value of calcium. When you go just above 0.773, the oscillations have maximum amplitude and minimum frequency. As you keep increasing VPLC, the amplitude of the oscillations goes down and this frequency goes up. They get faster and smaller. And then suddenly, when VPLC is equal to 3.75, they disappear. So it's just sort of an interesting circuit. Okay. Now, what we're going to do, and if you like, we could do this together. Create a new simulation. Maybe, well, given the time, I think I'm going to demo this and then we can decide whether you want to do it with me or just download the result. We're going to, now we have the actual basic model, that signaling that oscillatory calcium model. And we're going to do the same thing that we did before with the delta notch model. We're going to take that model, we're going to put it into the cells, and then we're going to have to couple neighboring cells because we're going to have to get the calcium from one cell to another. And so in this case, we would create a new project. We'd create a simulation of 256 by 256. In this case, we don't want periodic boundary conditions. We use that fluctuation amplitude 10. I like pixel copy range of 2. We'll use a rectangular slab initial layout. We still only have one kind of cell, although we might eventually use two kinds. No chemical fields, and we'll have contact energy, volume, flex. We don't have cell growth in this model. We'll have neighbor tracker turned on, and that's all we have. So do people, maybe it's not so bad. So maybe why don't we people walk through that? Okay, let's do this together. Okay, so create a new simulation. With Wizard. Create a new simulation. Make it 256 by 256. The default are no flux boundary conditions, so you get that one for free. Change the pixel copy range to 2. And then rectangular slab initial condition, which is also the default. Create a single cell type called cell. And then turn on contact, volume flex. Sorry, volume local flex, not volume flex. So we do have to change that one. And then cell neighbors for tracker. Okay, did that work? And then create the simulation. Go to the Twitter to the steppable. Sorry, go to Twit. Go to the XML and change the box mid to 51 by 51 to 204 by 204. Okay, got that? Okay. And now in the steppable, you're going to have to define the initial size of the cells. So at the very beginning of your Python code, you can create volume, coefficient, the lambda volume and target volume, 6 and 50. And then in the start function for cell and self.cell list, set all the cells to target volume and lambda volume. Okay. And we're going to have to change the adhesivity just a little bit. In the XML, we'll make cell medium, doesn't matter. Make cell medium energy of 12, cell of 6. Okay. And then we want to run that simulation. It's not going to do anything very interesting. You'll get that. So did that work for people? Anybody? Emma, did that work?
Yes.
Great. Okay. So now we're going to load cells into we're going to load that Zartmann model into the cells. And some of the cells are going to be called initiators, and they're going to have VPLC equal to 1.5. They're going to oscillate spontaneously. And other cells are going to be called standby cells. They're going to have VPLC equal 0.5. And then we have to couple the state dynamics of the cells and also the cell properties. So what you want to do now is you want to cut and paste that whole model that we had in the first simulation into this simulation. You put it at the very beginning of your Python so that you have it available. So just grab that model and paste it in to your simulation. Okay. And now in the start function, when we loop over those cells, we have to load that model into each cell. So we'll do self.add antimony to cell, model string, Zartmann model, which is what we called it, model name, Zartmann model, cell equals cell, because we're looping over all cells anyway. And then the step size, I didn't emphasize that. The step size is going to be given here. The step size is just 1 second per Monte Carlo step. And so you could make the step size just 1.0. Here I've created a parameter to hold it. Okay. And then in the step function, you have to integrate the ODE model. So you have to add a time step. And then we probably want to create some parameters that are going to define how many initiator cells we have versus responders. And so I create a variable which is probability of being an initiator, 0.1. The VPLC level for the initiators, 1.4, 1.5. And the VPLC level for the responders, which is 0.5. And you could use a shorter variable name if you don't want to type all of that. And then in our start function, after we have loaded the SPM model, the antibody model into each cell, we can roll the dice. We pull a random number. If NP random.random is less than the probability of initiation, 0.1, we change the value of VPLC to that higher value, VPLC initiator. And if you want to be fancier, you'd say else cell.sbml Zartmann model VPLC equals VPLC responder or standby. right? All right. We'd like to have some tracking fields. So we want to create our tracking fields for C, R, and P. And in the step function, we want to copy the values of C, R, and P into the cell dictionary. Again, you can load the final value, the final code if you want. It's not a terribly fast simulation. It takes a while for it to do anything. So what you'll see, is that the initiator cells oscillate, but they also oscillate in phase, and the standby cells don't do anything at all. And the reason for this is that we haven't coupled the cells together. Each cell is running as an independent oscillator. So to go further, we actually have to couple the calcium and IP3 between neighboring cells. And we also have to randomize the amount of calcium in the cell to begin with. And so, and this one I think we're not going to do hand. I'll ask you to just pull in 4.1.13. What we're going to do is the following. We are going to define the calcium flux. And this is a place where actually it would be nice if CompuCell had what's called Fickian diffusion built into it. There is something called an advection diffusion solver in CompuCell, and you can use it in this particular context. But it's a little bit finicky to use. Instead, what this code is going to do, and I'd ask you to download it while I'm explaining it, is it's going to measure the amount of calcium in each neighboring pair of cells. And it's going to say that we're going to move calcium between the two cells to try to equal out the calcium levels. So calcium will flow from the higher concentration cell to the lower concentration cell. And the rate at which it flows is going to be proportional to the difference, because it's diffusive, times the contact area between the two cells, times Fc, which represents the density of ion channels, the density of gap junctions in the cell. And we're going to do that both for calcium and for IP3. Both of them are transferred between neighboring cells. And so in this case, Fc and Fp represent the density of channels that allow the flow of these two molecular species between neighboring cells. And this is basically representing the diffusion of calcium and IP3 between neighboring cells. And when you do that, in this case, it's actually fairly simple to do it. We define a flux in each cell. We do that when we create the cells in the first place. And then at each time step, we calculate the common surface area between the neighbors, just the way we did for the delta notch model. And the flux is defined to be the amount in the neighbor minus the amount in my current cell. And I add that up. And that gives me the flux of calcium and the flux of IP3. And then, and I apologize for this misformatting of this slide. Somehow, again, the fonts got messed up a little bit. There's a flux conversion factor that says that the total amount transferred depends on the flux concentrate, the flux factor that we've just done, times this, basically the channel density. And so why don't people load exercise 4.1.13 and run it and tell me what you get. And I have a comment that this may run sort of slow. It takes about 1,000 Monte Carlo steps before anything happens. But at about 1,000 Monte Carlo steps, one of those initiator cells will start firing. And when it fires, you should see transmission of calcium spiking to the next and the next and the next cell. You have to visualize the chemical field. So you have to turn on the visualization of CP and I for it to do anything.
How much of what actually runs for CompuCell is Python and how much is C or something?
Well, it depends a little bit on what you're doing. If you're running if most of your setup is done in the XML, and the only things you're doing are display and cell growth and division in the Python, then probably 90% of it's in C++.
Explain why it's so fast. Hey Emma, you're seeing spikes. So I really want to do this for you because your interest is in the neural activity, and I really want to give you a propagating wave problem to show you that you really can do this kind of thing in CompuCell. As I say, I apologize a little bit for the fact that it's not a true action potential, but the mechanism of ion gated ion channels and storage release and continuous pumping, it's the same. It just happened to go to slower types. Okay, Alex, did it work? Did it ever spike for you?
Yeah, it is.
Great. Okay. So we have to wait. Wait. It'll be about to 2,000. So you see P is high in those couple of cells. P is high in those couple of cells. If you look at the maximum value of P, you see it's creeping up. This is auto-scaled, so red is changing its value. There you go. Now you're getting a wave. So you're getting this wave of calcium. Calcium in one cell induces a spike in the next, which induces a spike in the next, which induces a spike in the next. Why do you not get continuous spiking? Well, once the calcium's all released from the endoplasmic reticulum, you can't spike. Everything settles down, and you have to pump all the water back uphill for it to fall down again. And the repumping of the calcium back into the endoplasmic reticulum takes a while. And so you see that whole spike takes, what, about 200 time steps. And now we're back in the neutral region. And again, looking at that red at the top bar on your color chart, see it's 1.5, 1.6. And it'll take a little while for it to get up to the maximum, and then it'll fire again. And you'll notice again, if you look at the other cells, those bystander cells are not going to do much. You're not going to see their color change. They're staying down at 0.4, and so let's let it fire one more time. Now, we didn't walk through in detail what that first exercise was, where we looked at the frequency of oscillation and the amplitude of oscillation versus the parameter values. But what we could do is, reading off those frequency versus parameter values, we could tune this so that the oscillations were faster or slower, bigger or smaller. So we could change the interval between which we get firing. We could change the width of the pattern that's moving through. We can change the time scales and the length scales by essentially playing with that VPLC minimum and maximum value. So that's great. Thank you, Alex. That's fun. The last thing, which I will leave for you to try on your own, is that when calcium fires, it actually causes the actomyosin in the cell at the top of the cell to contract. And so it's very easy to add one line to your code. If you have the code running, you can add one line to your code, which is at each time you say V target for the cell is whatever its natural volume is, divided by 1 plus the calcium concentration divided by it happens to be 1.2 is a good number to play with. And then what you'll see is that as the wave goes through, you'll get a contraction of the tissue just in the region of the wave. Now, if we had some other things going on, we could actually have this tissue crawl in space. In reality, it doesn't do that. And it's not clear that this contractility is actually functional in the embryo, in the Drosophila embryo. But there are other organisms in which this kind of pulsatile contraction is very important. Your heart and the contractility of the myofibers in your heart basically works in the same way. And the individual myofibers contract spontaneously. This frequency of each individual myofiber in your heart by itself is not quite the same. However, when you put the fibers together, they couple so they synchronize, so they contract more or less synchronously. But the contraction is not over large distances, is not uniform enough. And then that contractility itself is modulated by the neurons that innervate the heart, that change the frequency of the oscillation to keep the whole heart synchronization. And you can build models like this of the heart, where you can simulate things like in the heart, you don't want those initiators to be doing that. You don't want multiple initiators. If you do, you have fibrillation. And so you can build models, for example, of fibrillation in the heart and various kinds of heart failure, basically using the same kind of model that we have here. And so if we do this apical contractility model, you basically just add one line. Cell.target volume is whatever the normal cell target volume is divided by 1 plus the Zartmann C divided by calcium volume coefficient. In this case, I set that to 1.2. And you'll see that you'll see the waves propagate and you'll see the tissue contract. And so that's a fun simulation to play with. Because of the fact that you're on a lattice, the tissue as a whole won't crawl. If you did this simulation in a dish, the tissue would actually walk on the substrate. But here, it's pinned to the lattice. We don't have a substrate for it to walk on, so the center of mass of the tissue won't move very much. You'll see the tissue change shape, relax, change shape, relax, but it won't move. The center of mass won't move. But as you play with it, you could get also mechanical coupling. If you make alpha smaller, the contraction is bigger. And you can play with the way that affects the patterning. And so that's a sort of a fun game to play. Okay. For the last part of the talk, I'm going to just talk. This is going to be slides that are from Giulio Belmonte. You'll see the logo for North Carolina State on the bottom of the slides because I don't want to take away from the fact that he did this great work. All of the things we've done so far, we've talked about spatial variability in chemical fields. But we know that cells actually have quite complex shapes. And we've talked about the endoplasmic reticulum as an organelle. We know the cell has a nucleus. The nucleus actually has rather different mechanical properties from the cytoplasm. If you're a plant cell, you have chloroplasts in the cell. You have mitochondria in the cell. The cells have different regions. The cells may have an apical region. They might have a lamellipodium or philopodia. And so you might want your cell to have subdomains that have different mechanical or chemical properties from the main part of the cell. And so CompuCell doesn't allow you to build super complicated cell shapes. I'm sorry, no Purkinje cells in CompuCell. I mean, I suppose you could try to simulate one Purkinje cell, but that would be wouldn't be the best way to do it. But we can build cells which have more complicated structure. And a typical thing we might want to do would be to have a cell that had a nuclear compartment, a cytoplasmic compartment, and then we, a long time ago, talked about simulating membrane which had particular flux properties for controlling the diffusion of chemicals in and out of the cell. And so it's pretty common to have a cell with a nuclear compartment and a membrane and a cytoplasmic compartment, sometimes a membrane compartment. Cells are often organized in epithelia, sheets where the bottom surfaces of the cell have one set of properties. They stick to the substrate, to the basement membrane. The intermediate parts stick laterally to each other, and often the apical surfaces don't like to stick to anything. They often are responsible for secreting or absorbing materials from the outside world. In your gut, they would secrete mucus or absorb nutrients. In your lungs, they would absorb oxygen and secrete mucus as well. And so epithelial organization is definitely associated with the cells not being isotropic. And when we're going to do epithelial organization, very often we'll define an apical domain, the top domain of the cell, a basal domain which sticks to the substrate, and a lateral domain that sticks to the neighbors. And Alex has been working on doing this all semester, so this is a very familiar thing for him. One of the things that you could do when you build compartmental cells is you could build more complicated architectures. This is an old simulation, which I wish we'd developed more. The eye actually develops initially from a single layer of cells, where you get a group of cells that bud out and then pinch off and form a vesicle on top of that initial layer. And it's possible to simulate that process. It's called extravasation. It's also possible to simulate things like bacteria, which are long and rod-shaped, by dividing them, making them essentially beads on a string. And there's some beautiful old simulations from, goodness, 2007 now, by Jörn Staros. Actually, I'm going to be talking to tomorrow morning, where they did simulations of swarming mixing bacteria that way. We've done this, for example, in conjunction with people at the School of Medicine to simulate the tubules in the kidney. In the kidney tubules, there's active pumping of blood from the blood and then also into the urine. So you have to have pumping on both the top and the bottom surfaces of the cells. And you have to maintain the structural organization. In this case, it's a little more sophisticated because the polarization isn't predefined. It happens spontaneously. We probably won't have time to do that next week, but if we had a little more time, we could actually model that as well. You could also use this kind of simulation again in the Drosophila case, in that imaginal disk for the wing. There is a spontaneous polarization in plane called planar polarity, which is associated with particular molecular species which like to be next to each other between neighboring cells, just like delta notch. In this case, the molecules are called frizzled, Van Gogh, disheveled, and flamingo, prickled. And they will spontaneously organize to polarize the cells to make a consistent pattern of organization of the cells. In this case, the result of that is that they're a little, they're called hairs, but they're not really hairs in our sense. They're little cilia, or essentially cilia that stick out of the cell, individual cell. They're subcellular organization, and those are oriented in a particular direction. They change the aerodynamic properties of the wing, and they also sense air currents on the wing. And if you look at a microscopic level, they're all parallel to each other on the wing. So let me just go through a few of the concepts that we're going to have to be able to build compartmental cells. The most important thing that is different in a compartmental cell is the idea of contact energy. Suppose that we have two cells which have compartments in them. Suppose we have one cell and it has three subcompartments. Well, if we want those things to stay together, they have to adhere to each other more than they adhere to the medium, because we know if they adhere to the medium more than to each other, they'll spread out and dissipate in the medium. And so we have to define these subcompartments as cohesive to each other. In this case, to get the picture we have on the right, we would need the yellow cells to like to be surrounded by the cytoplasm. The nuclear cells like the compartment to be surrounded by cytoplasm. So it has to have a low contact energy between nucleus and cytoplasm. And L is lamellifolium. It has to like touching the cytoplasm. So it has to have a low energy with cytoplasm, relatively high energy with nucleus, and it likes touching the medium. So it has to have a relatively low energy with the medium. However, if I define that and I bring two of those together, well, if nucleus likes touching cytoplasm, and I bring two cells together, the nucleus will be perfectly happy to touch the cytoplasm of the other cell instead of my cell. And this will turn into a big glob. And so I have to define different contact energies between the subcomponents of a single cell and the same subcomponents when they're in different cells. That's not a very hard concept in reality. In reality, of course, it wouldn't work quite like that because there'd actually be a cell membrane that would keep things separated from each other. But we don't have the membrane. We have to create something that behaves like the membrane. So that's how we're doing it. And so what we're going to do then is, instead of having one table of contact energies, we're going to have two tables, one of which says, what are the contact energies between the components within a cell? And a second table that says, if the components are in different cells, how do they interact? So, for example, nucleus will like to touch cytoplasm in its own cell, but not like to touch cytoplasm in a neighboring cell. Lamellipodium will like to touch nucleus in its own cytoplasm in its own cell, but not cytoplasm in the neighboring cell. And so I'm going to have two tables of contact energies, and I'm going to have to adjust the contact energies between compartments within the cell separately from the ones between cells. And that's going to be the key concept that we're going to have to use again and again when we deal with compartmental cells. And I'm going to skip the details here. But we have to think about this pretty carefully. So typically when we're going to build simulations of this kind, we start out by saying, what are the energies within a given cell that we need? We'll do the internal contact energies first. We want this component of the cell to touch this one to repel this one. This one doesn't want to touch medium. The nucleus should stay in the middle, so it should be repelled by medium, and so on. And then we have to think, if we bring two cells together of the same type, how should the compartments interact? Well, cytoplasm to cytoplasm and neighbors, we might want the cells to stick. But we definitely don't want nucleus in one to stick to nucleus in the other, or nucleus in one to stick to cytoplasm in the other. And so we do have to reason those hierarchies out. And here's an example. We want nucleus in one to have a high contact energy with cytoplasm in the other, lamellipodium and nucleus in the other, because basically we want the nucleus not to touch anything in the neighboring cells. And the way CompuCell implements this is a little bit backwards. And the reason that it's a little bit backwards is historical. Compartments were added later. And so the cell ID doesn't change. The cell ID basically is a cell ID, but now it represents the compartment. And we define a super layer, which is a cluster ID that says we're going to group these cells, quote cells, that's why they're called generalized cells, into a single bigger object, which we're going to call a compartmental cell. And so if we had 1, 2, 3, and 4 in our picture on the left, and we say those are actually subcompartments of a single cell, we would define a cluster ID and say that 1, 2, and 3, and 4 are all part of the same cluster. And so the terminology may be a little bit different. This is what it'll look like. Here we have two compartmental cells. The nucleus of the top cell is cell number 59. The cytoplasm is cell number 88. And the lamellipodium is cell number 85. But they all have the same cluster ID 10. And it would really be nice if the cluster ID were named cell ID, and the cell ID were named subcell ID or compartment ID, but it's not. But basically, it's pretty easy. We define the components, and then we say all of these components belong to the same cluster, and that makes them a cell. Okay? The other thing that we're going to have to find out about is that when cells divide with compartments, it's a little bit more complicated. We're used to the cells dividing by the cell ID, but now we're going to have division by cluster ID. And so there is a separate mitosis stepable called mitosis stepable cluster base, where instead of passing the cell ID to the division algorithm, we pass the cluster ID to the division algorithm. And otherwise, it has the same options that we have for normal cell division. And it tries to do all of the same things. In this case, we'll use cluster volume. And one problem is that that's not actually something that's available. We have to figure that out for ourselves. So we have to determine the cluster volume by adding up the component volumes. But it will try, cluster division will try, here's the cell. And when we do cluster division, it will try to copy the subcellular architecture when it does the division. Now, you can see that when we do that, there is really no unique way to do that. In other words, we're jumping. In other words, if I have that cell with nucleus, cytoplasm, lamellipodium, and I cut it in half, there isn't really an obvious way to take that initial structure and impose that on the successors. And CompuCell will do its best to actually cut things in a reasonable way. And if the cell architecture is something like an epithelium with 3 layers, top, middle, bottom, and you're dividing it this way, it'll do it pretty well. Sometimes if you have very complex cell shapes, and especially if they're small, it will screw up and you'll get a cell which is missing a compartment or where the compartments don't do what you want. And so you're a little bit on your own when you get to very complex cell shapes. But that basic ability to do cell division, first to define subcompartments, and then to be able to do complex cell architectures is pretty interesting. Something else that you have to do by hand at the moment is, for example, in the case that we just did, where we had contraction of the cells due to calcium influx. In reality, we modeled those cells as two-dimensional. In reality, those cells are epithelial cells that are vertical. They're sitting like this. And in that picture, we're looking at the top of those cells. And when we have calcium-induced contraction, what's happening is the top layer is contracting, but the volume of the cell isn't really changing. What's happening is the volume of the cell is getting moved vertically down from the top into the bottom. And so if we were going to do that contraction in 3D, instead of having the volume of the cell shrink, what we would have to do is move target volume from the top compartment into the bottom compartment, then back up, keeping the total volume of the cell the same. And if we do that in 3D, actually, we get much more realistic movement of the tissue. You'd see that contraction wave would behave somewhat differently. It would be a lot smoother and would be a bit more realistic. It would also take longer to run because you're in a 3D simulation. And so we're not going to have time today to do any real exercises based on this, but I did want to introduce the ideas here and some of the things that you could do with compartmental cells. And again, you could use these for structural or functional heterogeneity. You could represent real things like the nucleus or conceptual domains like apical and basal domains. And the next time, what we'll do after people have done initial presentations is we'll create this epithelial sheet, and then I hope we'll be able to try to make it bend. We won't be able to get that full eye simulation done, but I'd like to be able to show you how to get some structural reorganization. And so that's where I'm going to stop for today.